Lecture number 26, gamma glutamyl transferase, albumin and prothrombin time. Gamma glutamyl transferase, the reaction is catalyzed by gamma glutamyl transferase is the hydrolysis of the gamma glutamyl bonds, which catalyze the transfer of gamma glutamyl functional group from molecules such as glutathione to any acceptor molecule that may be an amino acid, and thus a glutamate is formed. You can see here from the glutathione the group is transferred and the gamma glutamyl group is transferred. The acceptor is the amino acid. Gamma glutamyl amino acid is formed. Here the enzyme is gamma glutamyl cyclotransferase, and here is the gamma GT, gamma glutamyl transferase, and now in this way the enzyme glutamate is formed. This enzyme transferase is found in the cell membrane of many organs throughout the body, and highest concentration is found in liver. Release of enzyme in circulation, cell damage induced by disease or drugs. Increase the membrane permeability, causing the enzyme to spill into the sinusoid and then to the peripheral blood. So whenever there is cell damage or there is any disease of liver and or there is some cell damage by the drugs, then the increase in the membrane permeability is there, and in this way the enzyme is released into the sinusoid and then into the peripheral blood. Fragments of hepatocyte membrane are rich in gamma GT and bile acids. They act as detergents, solubilize and release the gamma GT from the plasma membrane. So, what is the function of bile acid? To solubilize and release the gamma GT from the plasma membrane. Here you can see the hepatocytes and the sinusoid. Here you can see a waving margin, and this shows that the cells are present in the sinusoid, and these are known as the sinusoidal cells. These are the hepatocytes. Functions. It is involved in the transfer of amino acid across the cell membrane and leukotriene metabolism. Peptide and protein synthesis, regulate tissue glutathione level. What are leukotrienes? Leukotrienes are the compounds made from 20-carbon fatty acid, unsaturated fatty acid, which is known as the arachidonic acid, which activates the immune and inflammatory responses. They are required for healthy immune reaction. Leukotrienes, they are very important for providing the immunity, and they are required for healthy immune reactions, and they attract and activate white blood cells to prevent and resolve the disease. Gamma GT elevation. Gamma GT is elevated in the blood in most diseases that cause damage to the liver or the bile duct, since they are very much found in the epithelial cells of the bile duct. So, gamma GT is raised in the diseases of liver, biliary system and pancreas. It is similar to alkaline phosphatase, but alkaline phosphatase is the first enzyme to be raised in the biliary disease. Although the function of both of them, origin, you can say that both of them is in the same condition, but still ALP, alkaline phosphatase, is the first enzyme to be elevated in the biliary diseases. After we have discussed the bilirubin, we discussed the liver enzymes, and now we'll discuss the albumin. Albumin synthesis is an important function of the liver. Approximately 10 grams is synthesized and secreted daily. With progressive liver disease, serum albumin level fall, reflecting its decreased synthesis. Since liver is diseased now, therefore the synthesis of the albumin will be decreased. And albumin concentration correlate with the prognosis of the chronic liver disease, because as the liver is damaged, the synthesis of the albumin will be decreased. But when the disease is cured or the liver retains its original function, then the albumin synthesis increases. Albumin levels are dependent on number of other factors also: nutritional status, catabolism, hormonal factors, urinary and gastrointestinal losses. यानी किडनी डिजीज है तो उसमें यूरिन के थ्रू इसका लॉस होगा एल्बुमिन का. Then we come to the prothrombin time, the synthesis of the coagulation factor, except factor 8, is an important function of the liver. Prothrombin time, it measures the rate of conversion of prothrombin to thrombin, requiring factor 2, 5, 7 and 10, and thus reflects a vital synthetic function of liver. Here you can see prothrombin is converted to thrombin, and thrombin then converts fibrinogen to fibrin, which is actually the blood clot. Hepatocyte and liver sinusoidal endothelial cells are involved in the synthesis of the coagulation factor. Liver sinusoidal endothelial cells, which are specialized endothelial cells that line the liver sinusoids, have been reported to participate in a variety of liver functions such as blood macromolecule clearance and factor 8 production. So liver, what is a function of the liver sinusoidal endothelial cells? They perform basically two main functions. They clear the blood from the macromolecules, from the extra amount of the molecules which are present in the blood, as well as the production of coagulation factor 8. Here you can see the sinusoidal cells. High prothrombin time usually means that there is some serious liver damage or cirrhosis. The liver is not making the right amount of the blood clotting proteins, and so the clotting process takes longer. The prothrombin time is prolonged in vitamin K deficiency and liver diseases, since vitamin K is required for the gamma carboxylation of many coagulation factors. It is important to distinguish prolonged prothrombin time due to hepatocellular disease from chronic cholestasis with that of fat malabsorption. यानी prolonged thrombin time जो है वो hepatocellular disease की तरफ से है या की वजह से है या फैट malabsorption की वजह से है. तो इसमें क्या करेंगे कि vitamin K is given to the patient. If it is due to chronic cholestasis with fat malabsorption, तो problem dissolve हो जाएगी. ठीक है ना. It will reduce the prolonged prothrombin time. The prothrombin time will be reduced and the problem will be dissolved. But अगर ये liver disease की वजह से है, so the vitamin K cannot cure this. But if it is due to the intrinsic liver disease, the vitamin K will not reduce the prolonged prothrombin time. So the prolonged prothrombin time may be due to two cases, two reasons: hepatocellular disease or chronic cholestasis with fat malabsorption due to the fat malabsorption, right? And it can be differentiated by the administration of vitamin K, which will reduce the prolonged prothrombin time, and it can resolve the problem. But if there is any liver disease, intrinsic liver disease, so vitamin K will not work in reducing the prolonged prothrombin time. Common liver test associated condition, here are the enzymes: AST, ALT, hepatocellular injury, which may be due to ethanol, hepatitis, ischemic injury, acute biliary obstruction. In all cases, ALT and AST will be raised, whereas alkaline phosphatase, it will be raised in cholestasis. Children during bone growth, because bone growth, one of the fraction of the alkaline phosphatase, it comes from the bone disease. Pregnancy, because one of the isoenzyme is of placental origin. Then gamma GT, cholestasis, medication, ethanol toxification, anorexia nervosa, hyperthyroidism. Bilirubin, it is raised in all acute or chronic liver diseases and congenital disorder of bilirubin metabolism.